Spermatozoa. The sperm is about 60 millimeter long and is a long single cell which has a head and a tail. The head is the nucleus of the cell containing DNA material. Head is capped by acrosome made up of mucopolysaccharide and acid phosphatase enzyme. The acrosome is covered by a membrane called as acrosomal membrane. In the fallopian tube, acrosomal membrane is removed so that the acrosomal enzymes are exposed. These enzymes are digestive in nature that facilitates their activity by enabling the sperm to penetrate the ovum to cause fertilization. The removal of acrosomal membrane is called as sperm capacitance. The highly elongated structure of sperm enable it to swim forward in the female genital tract at the speed of 3 millimeters per minute. Sperm reach the uterine tube 30 to 60 minutes after the copulation. So this is the structure of the sperm, which has a head, neck, middle piece and the principal piece. And the head has a nucleus with acrosomal cap, whereas middle piece has abundant mitochondria. Semen. Semen is the fluid that is ejaculated at the time of orgasm. It contains sperms and the secretion of seminal vesicles, prostate, Cowper's gland and urethral glands. You see the composition of the semen. Uh, uh, these are some of the substances secreted from the different structures. Like seminal vesicles, it produces phosphorylcholine, ergothioneine, ascorbic acid, flavins, and prostaglandins, whereas prostate gland produces spermine, citric acid, cholesterol, phospholipids, fibrinolysins, fibrinogenase, zinc, and acid phosphatase. And they have a plenty of buffers like bicarbonate and hyaluronidase. The specific gravity of the semen is 1.028 and pH is between 7.35 to 7.5. The average volume per ejaculate will be around 2.5 to 3.5 ml. Sperm count is around 80 to 120 million per ml, with fewer than 20% of the abnormal forms. Seminal vesicles contributes to almost around 60% of the total volume of the semen, and prostate gland around 30%, 20 to 30%. Note that clotting enzyme from the prostatic fluid causes the fibrinogen of the seminal vesicle fluid to form a weak fibrin coagulum that hold the semen in the deeper regions of the vagina. The coagulum then dissolves during next 15 to 30 minutes because of the lysis by fibrinolysin formed from the prostatic profibrinolysin. Nutrition to sperm is mainly provided by the fructose, and fertilization is enhanced by the prostaglandin present in the seminal vesicles. Coming to Sertoli cells. Sertoli cells are large, complex glycogen-containing cells that stretch from the basal lamina of the tubule to the lumen. Some of the functions of Sertoli cells are: Sertoli cells secrete androgen-binding protein, inhibin, activin, and Mullerian inhibiting substance, that is MIS. They have receptor for the follicular stimulating hormone and testosterone. After combining with FSH, Sertoli cells stimulate the first half of the spermatogenesis. Subsequently, testosterone Sertoli cells binding causes development of the last half of the spermatogenesis. Sertoli cells are components of blood testis barrier, and Sertoli cells they produce estradiol from testosterone. They also provide nutrition to the germ cells, and they also help in conversion of testosterone into 5-alpha dihydrotestosterone. Sertoli cells and epithelium of the epididymis secrete a special nutrient fluid that is ejaculated along with